हाय, वेलकम टू थिंग्स ऑफ रियलिटी। कोरोना वायरस को लेकर अमेरिका से एक गुड न्यूज आया है। अमेरिका में एक रिसर्च इंस्टीट्यूशन है, यूनिवर्सिटी ऑफ पिट्सबर्ग, वहीं पर कोरोना वायरस का एंटीडोट या फिर वैक्सीन डेवलप हो चुका है। इस वीडियो पे हम यही डिस्कस करेंगे। उससे पहले आप इस चैनल को सब्सक्राइब कर लीजिए, यहीं पर आपको रियलिटी से जुड़कर सभी वीडियो मिल जाएगा। यूनिवर्सिटी ऑफ पिट्सबर्ग साइंटिस्ट का कहना है पीट गो वैग नाम का एक वैक्सीन उन्होंने डेवलप कर लिया जो कोरोना वायरस को पूरी तरह से खत्म कर देगा। जैसे 2003-04 में सार्स वायरस एंड 2012-13 में मार्स वायरस को खत्म किया है, वैसे ही 2020 में कोविड-19 को पूरी तरह से खत्म कर देगा। सार्स एंड मार्स वायरस वो भी एक पूरा कोरोना वायरस है, लेकिन 2020 में जो कोरोना वायरस आया है उसका स्पाइक प्रोटीन थोड़ा सा हाई। जैसे मानव शरीर का ग्रोथ के लिए प्रोटीन जरूरी होता है, वैसे ही वायरस का ग्रोथ के लिए प्रोटीन जरूरी है। पहले जो कोरोना वायरस था उससे भी ज्यादा ताकतवर 2020 में जो कोरोना वायरस आया है, इसलिए जल्दी से जल्दी बहुत लोगों के अंदर ये वायरस फैल जाता है। इस बारे में यूनिवर्सिटी ऑफ पिट्सबर्ग साइंटिस्ट का क्या कहना है, पहले वो सुन लो, फिर मैं आगे की कहानी बताती हूँ।
Question. So the 2003 SARS-1 vaccine, the critical aspect of that study was that help us to identify what is the target for a coronavirus vaccine. And at that time, we determined that the spike protein that now everybody uses as a target for this coronavirus is the best target for a coronavirus vaccination. The second study on the MERS vaccine, the other coronavirus that emerged few years ago, we test different platform, and in 2014 we determined that the subunit protein vaccine was as good as other genetic systems. So those two vaccines teach us how to translate all this finding in the new SARS coronavirus 2 vaccine.
Okay, thank you. Next question.
2003 से ही कोरोना वायरस को लेकर रिसर्च चल रहा है, इसलिए कोरोना वायरस का वैक्सीन डेवलप करना बहुत ही आसान हुआ। लेकिन कोविड-19 का जो वैक्सीन है पिटगोवैक, वो अभी तक सिर्फ चूहों में ही टेस्ट हुआ है। पहले 100 चूहों को आर्टिफिशियल तरीके से तैयार कोविड-19 वायरस से संक्रमित किया गया, उसके बाद लैब में तैयार पिटगोवैक नाम का वैक्सीन दिया गया। रिजल्ट पूरी तरीके से चौंकाने वाला था। 100 चूहों में से 100 चूहों पूरी तरीके से ठीक हो गया और इस वैक्सीन का कोई भी साइड इफेक्ट नहीं हुआ। अभी तक रिसर्च सिर्फ चूहों में ही हुआ है, इंसानों के ऊपर रिसर्च अभी तक चल रहा है। इस वैक्सीन को इंजेक्शन के फॉर्म में नहीं दिया जाएगा। इस वैक्सीन को देखने में एक बैंडेज की तरह, बैंडेज जैसे आपका शरीर में लगता है वैसे ही इस वैक्सीन को बैंडेज की तरह आपका शरीर में लगाना पड़ेगा।
With a novel technology that's referred to as a dissolvable microneedle array. What the microneedle array is able to do is to deliver the vaccine antigen directly into those areas of the skin that are made to make an immune response. And so it results in a very potent vaccine. When you're thinking about how these microneedle arrays are actually used in practice, think about them as almost like a Band-Aid. And so the microneedle
यूनिवर्सिटी ऑफ पिट्सबर्ग साइंटिस्ट का कहना है इस वैक्सीन को बनाने का प्रोसेस बहुत ही आसान है। करीब एक दिन में एक आदमी करीब 100 वैक्सीन बना सकता है। अगर मैं इंडिया की बात करूं, इस वैक्सीन को पहले इंसानों के ऊपर रिसर्च होते होते करीब 1.5 से 2 महीने का वक्त लग जाएगा। फिर इसका मैन्युफैक्चरिंग प्रोसेस स्टार्ट होगा, फिर जाके इंडिया में आते आते करीब 4 से 5 महीने का वक्त लगेगा। इस वैक्सीन को लेके जो प्रेस कॉन्फ्रेंस हुआ था उसका मैं थोड़ा सा फुटेज आप लोगों को दिखा देता हूँ।
think you can? Yeah, thank you. That's a good question. So Dr. Fauci is exactly right. That is the time course for vaccine development. Much of that time course, and the reason there is such a gap in how short versus how long it could be, depends on how the vaccine develops through the clinical trial. So it depends on the level of responses that are seen as the trials are being done, the safety data that comes in during the course of the trials. And so for that reason, it's impossible to give a very precise timeline on when a vaccine would be ready. We are able to start the trial as soon as we have regulatory approval. In terms of scale, our vaccine platform is very conducive to scaling. And so we will be able to ramp up production using microneedle arrays very quickly because of the straightforward technology that's involved and because of the requirements for much lower doses of antigen that need to be produced for each vaccine.
Thank you. Next question, Bill O'Toole from Pittsburgh Quarterly. Bill, go ahead.
Am I coming through? Hello? Hello?
Oh,
Am I coming through?
Yes, you are.
yeah.
Yeah, a related question. How do you see manufacturing working practically if your trials do indeed succeed? Would it just be individual labs all over the country making them? One big center? What would it look like?
Yeah, I suspect, first let me say that we haven't gotten there yet, that all of our focus right now has been getting this vaccine into clinical trials to evaluate safety as soon as we can. But the vision would be a centralized facility capable of making millions of these vaccines at a rapid rate.
Thank you. Again, to ask a question, go to the participants icon at the bottom of the page and press the raise your hand button. That will put you in the queue. I will then read your name and your line will be unmuted for you to ask your question. So just a reminder of how to get into the queue to ask questions. The next question comes from Danielle Fitzgerald of the Beaver County Times. Danielle.
Hi, everyone. Thank you so much. Yeah, I have a couple questions. I guess this might be more for Dr. Ely. But my question was, why are the number of positive tests so low? You mentioned somewhere around 10% of tests are positive. It feels kind of low for a highly contagious virus. Do you think we're testing the wrong people or using the wrong symptoms as a benchmark? And related to that as well, I heard that losing a sense of smell and taste or perhaps even a really more important identifying symptom. I don't know if that's true or not. I wanted to get clarity on that. Should we be testing folks more with those sorts of symptoms?
Those are great questions, and thanks for asking them. I don't know if 9 or 10% is low or high. We're still learning a lot about how the virus enters a community, and it doesn't always enter every community in the same fashion. You know that when you look both globally and even across the Commonwealth, that the amount of positive cases that are either symptomatic or admitted to the hospital varies dramatically across the region. What we know is we're seeing this essentially 9 or 10% positivity rate, and why that is, I couldn't speculate. What I can tell you is that our background surveillance suggests that it's not dramatically undercounting what's available out in the community, and you've heard about that research before. Your other question had to deal with, remind me now, the second question. Oh, smell and taste. Right. It is one of the unusual things about COVID-19 infection is that early on it does seem to disproportionately, for this particular infection, change people's sense of smell or taste. And taste is often related to smell, so sometimes it's hard to separate them out. And we've sent out reminders to our clinicians to look for that finding. It's not specific, but it certainly is something that should help raise your awareness about this potential. And we've seen people not only have those symptoms, but after recovery report improvement. So I do think it is part of the clinical picture, just like the respiratory symptoms, just like fever, that talented physicians, talented nurses, talented practitioners should be aware of and ask about.
Thank you. Next question comes from Fox 43 in Harrisburg. I believe it's Matt Maisel. Matt, go ahead.
Gentlemen, thanks for taking these questions. Two questions real quick. First, how long have you been working on this particular vaccine? When did you start with the testing? And also, people just in general and naturally are scared during this entire process. What do you tell them who might be watching this right now or might be watching later and are skeptical when they hear something that it may still be another year plus for something to be finalized?
So yeah, we start to work on this vaccine exactly January 21st. January 21st, the sequence of SARS coronavirus 2 became available. And with that sequence information, we were able to generate quickly. Took us 7 days to generate the actual vaccine. And then short after, we immunize the animals. So it's about 8 weeks that we are into this studies and January, the end of January.
Thank you. We'll take another question from Central PA. Carrie Pauling from northcentralpa.com. Carrie, go ahead.
Hi, thank you. I'm wondering whether or not there's a concern if there is an accelerated trial that developers could miss dangerous side effects or other negative reactions that people might have.
Thank you. That's a very good point. What you're putting out is that there's a reason that these trials take the length of time that they do. We are taking every precaution, and you'll see in all of the trials that develop over the next months that safety is our top concern. And that's why these trials are done so carefully and can take so much time.
Thank you. Again, we have time for just a few more questions, so go to your participants icon at the bottom of your page and press the raise your hand button. That will put you in the queue. Next question comes from Ted Lutz at the Kane Republican. Ted, please go ahead.
Yes, sir. My question has to do with the testing. Who should go for the test? And we live 90 miles from Erie, and that's where one of our participants did go, and they tested positive. But I see you up in Jamestown, New York, which is a lot closer. Should just anybody go to test, or what's your policy for accepting candidates for testing?
We prioritize the order in which people get a specimen collected and sent off. And at the top of that list are people who have symptoms, and particularly people who have symptoms and require hospitalization. That's where we can be not only most effective for their care, but make sure that people around them are the most safe. One group of people that we simply don't have the capacity today to test are people who have absolutely no symptoms, but maybe have a concern about having acquired the infection. It's not that that's unimportant, and at some point in time, we'll want to know what does the population that doesn't have any symptoms, how often do they have the infection? But right now, that's not the highest priority. So my advice would be seek testing if you have the symptoms: cough, fever, difficulty breathing, the smell and taste things we just talked about. And while you're doing that, make sure you remain isolated so that if you do have the infection, you haven't spread it to others.
Thank you. Next question is Oliver Morrison from Public Source. Oliver, please go ahead.
Thank you. I'm curious if the information that you're sharing in this publication is going to be available to scientists other places so that they can be testing it at the same time. And if so, is there going to be any advantage to sharing, given the public health issues, to sharing this information that might otherwise be a source of profit freely to other countries and other places so that it would be available as quickly as possible to the people who need it?
Thank you. That's an excellent point. With this current pandemic, we are all on board with everyone collaborating to attack the virus. We are all working together and sharing data very openly. The publication that's specific to our vaccine is right now available to all of the public. It's posted on the journal's website, and we are collaborating with anyone that we can help in terms of data sharing and sharing our experiences.
Thank you. Next question, Alex Weedhoff from the Cranberry and Butler Eagle. Alex, please go ahead.
I have two questions. The first one is, do we know what level of widespread immunity we will need to have by time the vaccine will be developed? And if so, do we know how that would be able to be deployed by vaccine, or if some people will naturally have it because they will have gotten the coronavirus already? And my second question is in terms of background monitoring. Do we have recent information, data on how many people we have tested showing up? I believe this was people showing up to the ER with flu-like symptoms, just being tested for that with not necessarily identifying data associated with that.
Well, I can take the second question. The surveillance data that you're talking about that's been presented at previous press conferences is coming near its close. There have been over 3,500 patients in the Western Pennsylvania area, both adults and children that have been tested. And again, it shows that beginning through the end of January, the middle of January through now, there was not an early presence, at least not widespread, that existed of the virus. And the pattern of the virus presence right now looks very much like the clinical picture that we're seeing. In other words, there's not a disconnect. And that's in a large but not exhaustive sample.
As far as the first question, so it is our expectation that as patients recover from the infection, they will have some level of immunity. Tests are being developed to specifically address that, including tests that are being developed here at UPMC. So we would expect some amount of what is described as herd immunity. The vaccine would be a supplement to that herd immunity.
Thank you, doctors. Next question, Todd Bartley from WEJS in Williamsport. Todd, go ahead.
Thank you. You had mentioned the potential of moving to a human trial. First of all, what's the timeline on moving to the human trial? How far away are we? And then how would you end up selecting the candidates to participate in the human trials?
Yes, so the timeline will be established by the regulatory process. From a clinical perspective, we're ready to go with and initiate a clinical trial as soon as we're able to meet the necessary requirements. The second part of the question was, sorry.
How are people going to be selected?
Oh, so we will be starting with healthy volunteers in the Pittsburgh area. Those will be the first cohorts of patients that enter the trial. And we will be basically looking at multiple doses during the course of the trial to find the most effective dose.
David Kaplan from WTAE has a follow-up question. David, go ahead.
Just talk a little bit about, we heard at the federal level just talking about the next 2 weeks. So big picture, I mean, are you all preparing for and or expecting some surge? And then my second follow-up question, can you talk about a little bit about the normal timing for peer review, the normal amount of time it would take to get the federal approval for a vaccine like this, and kind of, what's the normal timeline and how condensed do you all really hope that you can make this timeline to get things going in terms of clinical trials?
So the first question is, what do we think is going to happen, and when do we think the surge will become very high, and when will it hit its peak? I have no reason not to agree with some estimates that say the surge may be at its peak within 2 to 3 weeks. The truth is we don't know. There are other estimates that would suggest that it could last even longer than that. Our approach, and you've heard this before, is that we prepare for the worst, we hope for the best, but we let the experience on the ground guide what we're doing right now and how we prepare and deploy day in and day out.
So safety is the prime concern in most of these clinical trials. And that's the reason that the typical review process can take months. We're hoping with the expedited review and all the efforts that the regulatory agencies have made that we can reduce that regulatory process from months to weeks.
Okay. Thank you. Our final question comes from Jamie Martinez of the Tribune Review. Jamie, go ahead.
Hi. To just jump off of a question that was asked earlier related to testing, how would this vaccine then be deployed to the public? Would it go to hardest hit areas first? Would it make sense for it to go elsewhere first? Which areas would probably see it, be among the first to be receiving the vaccine?
So I think that when the vaccine is actually ready for widespread deployment, it would most likely be ready broadly so that we wouldn't have to ration the vaccine. I would expect that the manufacturing and scale up would occur during the testing process so that plenty of vaccine would be available if it proved to be effective.
Okay, thank you. And that concludes the time we've allotted. Thank you all.